Plasma proteins. Mainly consists of solid elements like red blood cells, white blood cells, platelets suspended in the liquid medium called plasma. The albumin, the name is derived from the white precipitate formed when egg is boiled. It is present in high concentration, that is about 60% of total protein in the albumin. It has one polypeptide chain with about 585 amino acids and 17 disulfide bond with a molecular weight of 69,000. The liver produces 12 grams of albumin per day. The albumin helps to maintain the intravascular colloidal osmotic pressure. This pressure prevents the flow of plasma into the tissue space by which the fluid volume inside the blood vessel is maintained. The decrease in albumin level leads to the escape of fluid into the tissue paper, resulting in the edema. The albumin is intravascular transporter proteins for many hydrophilic substances like unconjugated bilirubin, fatty acids, minerals, drugs, and vitamins. The albumin serves as a source of amino acids for tissue proteins synthesis, which is broken down. Albumin shows maximum buffering capacity. The large number of histidine residues present in the albumin is responsible for buffering action of albumin. The decrease in serum albumin is called as hypoalbuminemia, seen in the cirrhosis, nephrotic syndrome, and malnutrition. Globulin, the different types of globulin present in the plasma are alpha 1, alpha 2, beta 1, beta 2, and a gamma globulin. These proteins are glycoproteins with the molecular weight ranges from 90,000 to 130,000. The alpha and beta globulin helps to transport protein, hormones, vitamins, minerals, and lipids. The gamma globulin are known as a immunoglobulin, and they provide immunity against infections. Fibrinogen is an acute phase protein. The fibrinogen is an essential factor in the blood coagulation. The conversion of a fibrinogen to fibrin occurs by cleaving the arginine glycine peptide of a fibrinogen. It is synthesis in the liver. The fibrin monomers aggregate and precipitate to form clot. Different types of plasma proteins and their concentration in the blood as follows. Separation of plasma protein. The chemical and immunological methods are available that can quantify the concentration of a specific plasma protein with high degree of specificity. Less commonly, electrophorese is used to provide a semi-quantitative estimation of a pattern of serum proteins. Plasma protein can be separated by different techniques, which mainly depends on the certain properties of the proteins. Charge group, which are present in the protein, molecular weight of the protein, gel filtration column, which packs with the gel and used to separate the protein. The protein are separated depending on the molecular weight. Precipitation of protein by salt. Albumin is soluble in water, whereas globulin are less soluble in water. All proteins are soluble in dilute salt solutions. As the concentration of salt increases, the protein gets precipitated from the solutions. When the ammonium sulfate is added to a solution of a protein until it completely saturated, the availability of water molecules for protein is decreased, causing the protein to precipitate. This process is called as salting out. Albumin is precipitated to full saturation with the ammonium sulfate. Electrophoresis separates the protein into 5 broad fractions: alpha 1, alpha 2, beta 1, beta 2, and gamma. Each of the globulin fractions consists of a mixture of several proteins. Electrophoresis is the migration of charged molecules in electric field. Negative charged particles moves towards the anode, and positive charged particles moves towards the cathode. Protein is a solution of our plasma and can be separated from another by electrophoresis because the charged molecules. Protein contains a charge due to the presence of amino acids and a carboxyl group. The presence of large number of negatively charged depends on the number of CO minus group. If the protein has more NH3 plus the group, it will have more positive charge. At acidic pH, the protein will have more acidic charges, so protein will be positive charge and moves towards the negative electrodes in an electric field. At alkaline pH, the protein have more negative charges, hence the protein will have negative charge and moves towards the positive electrode in an electric field. At isoelectric pH, the number of positive charge is equal to number of negative charge. At this pH, the net charge on the protein is 0 and is electrically neutral. Each protein has its own isoelectric pH. For example, albumin has an isoelectric pH 4.7, globulin has a pH 5.8 to 7.3. Interpretation: this is done through several inspection of the band. The quantization of each band can be done with a densitometer. On electrophoresis, the plasma protein gets separated as distinct bands, namely albumin, alpha 1, alpha 2, beta 1, beta 2, fibrinogen, and gamma globulin. Electrophoresis of the serum samples will show all band except fibrinogen. estimation of plasma protein, determination of serum protein, albumin, globulin, and the ratio by Biorich method. Proteins belonging to different globulins. There are different types of globulins, and each of them constitute mixture of several proteins. Alpha 1 globulin, alpha 1 antitrypsin, alpha 1 acid glycoprotein, alpha 1 lipoproteins, thyroxine binding proteins, alpha 1 antitrypsin. It is an acute phase reactant and protease inhibitor present in the extracellular fluid throughout the body. The class of certain proteins in the blood may increases up to 1,000-fold in several inflammatory and neoplastic conditions. Such proteins are called as acute phase proteins. It neutralizes lysosomal elastase, which is released during the phagocytosis, and phagocytosis particles by polymorphonucleonucleoside. Thus, alpha 1 antitrypsin has a proteolytic role in the body. The alpha 1 antitrypsin level is increased during the any infections or inflammation because this protective role. Hence, it is known as an acute phase reactant. The alpha 1 antitrypsin level are found to be low in patients with genetic diseases. These patients suffers from respiratory diseases such as emphysema or liver damages. The decreased alpha 1 antitrypsin leads to loss of elasticity of lungs tissue, which results in emphysema and impaired ventilation and vulnerable to serious respiratory infections. Reference range newborn 145 to 270 milligram, adult 78 to 200 milligram. Estimation of antitrypsin and alpha 1 anti-chymotrypsin in serum. The proteolytic enzymes hydrolysis casein with the formation of a smaller peptides. The enzyme reacts after a suitable interval of time. It arrests by addition of TCA, which precipitates the protein, but the peptides are soluble in acids. TCA soluble fragments are measured of proteolytic activity of these enzymes. When the inhibitor is added to the pre-incubation mixture, it prevents the release of a peptide by the proteolytic enzyme. Thus, elimination of TCA soluble component in the presence and absence of inhibitor is measured independently against the proteolytic enzyme. The TCA soluble fragments were analyzed by the method of Lowry et al. The final color formed is a result of Biuret reaction and peptide with copper ion in alkali and reduction of phosphomolybdic phosphotungstic reagent by presence of tyrosine and tryptophan present in the treated peptide. Wilson disease. Alpha-globulin, the important proteins under this group are alpha macroglobulin. It has a protective role in body. It is synthesized by the hepatocytes. It activates all proteases. Therefore, it is considered as an in vivo anticoagulant. Hepatoglobulin. This binds with the hemoglobin and helps in the breakdown of hemoglobin to bilirubin. Ceruloplasmin. It is a copper-containing protein in plasma. It is known to have an antioxidant property. It is thought that the cellular plasmin is involved in iron metabolism. It acts. It is an acute phase reactant protein. Its level is increased in plasma in infection and malignant conditions, especially in Hodgkin disease. Cerebral plasmin level is decreased in Wilson's disease and used in the diagnosis of the disease. Wilson disease is a genetic disorder. It is a inherited autosomal recessive disorder. It is a disease of copper metabolism where copper 2 plus is accumulated in the liver tissue, brain, cornea, and kidney tubules. This results in liver damage, mental and retardation, and blindness. The peculiar findings of this disorder is that copper deposit has a green or golden pigmented ring around the cornea. This is also known as a Kaiser's Fraser ring. The renal failure may be observed in the patients because copper deposition in the kidney. The plasma copper and ceruloplasmin level are low in this condition. Normal serum levels 25 to 50 milligram per deciliter. Beta globulin. The important proteins of this classes are transferrin. It is a transport protein for iron in the plasma. It is a negative acute phase protein because its level decreases in some inflammatory condition. Beta lipoprotein, the carrier LDL cholesterol in the plasma from the liver to the peripheral tissue. C reactive protein. beta 2 microglobulin C-reactive protein. The C-reactive protein is an acute phase reactant. ERP is released in response to acute injury, infections, or inflammatory stimulus. The recent development of high sensitivity assay of CRP has enabled investigation of this marker of systemic inflammations. It consists of 5 identical non-glycosylated polypeptide subunit non-conventional non-covalently linked to form a disc-shaped cyclic polymer. This consists little or no carbohydrate and migrate both on cellulose or agarose electrophoresis anywhere from gamma to beta region. Clinical secondary its level rises more than 6 hours after triggering stimulus, peaks within 48 hours, short half-life of 5 to 7 hours. Indications of detection, monitoring, and differentiating conditions of systemic lupus erythematosus, CRP, higher in rheumatoid arthritis, Crohn's disease, ulcerative colitis, and pyelonephritis. The plasma CRP level increases after the stress, trauma, inflammation, infection, myocardial infarctions, and neoplastic disease. The reduction of CRP level is clinically useful for screening of organic disease, assessing the activity of an inflammatory disease such as rheumatoid arthritis, for detecting the intercurrent infections in systemic lupus erythematosus, leukemia, and after surgery, for detecting the renal transplant resistance, for managing neonatal septicemia and meningitis. Analysis radio immunoassay, analysis radio immunoassay, RAD, radio immunodivision, radio immunoassay, nephrometer, and homogeneous FIA. Reference interval as per RAD, the rare rate nephrometer is 80 to 800 milligram per deciliter. Immunoglobulin, the defense strategy. The strategy of a body or collectively known as immunity. The two types of immunity are in cellular immunity. This is mediated by the T lymphocyte and or T cells. The humoral immunity, it is mediated by the specialized group of protein known as immunoglobulin or antibody. The B lymphocyte or B cells are responsible for the production of immunoglobulin. The immunoglobulin are also known as gamma globulin. The protective infections function of antibody synthesis is responsible for to a foreign substance called antigen. Provide immunity. 5 types. Different types of immunoglobulin are IgA, IgG, IgM, IgD, IgE. Structure: all immunoglobulin molecules consist of two identical heavy chains, molecular weight of 53,000 to 75,000, and two identical light chains, molecular weight 23,000. They are held together by disulfide because heavy chains of molecular weight are linked to carbohydrate. Hence, immunoglobulin or glycoprotein. Each chains of IgG has two regions, namely the constant and variable. At the amino terminus, half of the light chain is variable region. At the carboxy terminal, half of the constant region. There are 5 types of heavy chains: gamma, alpha, mu, delta, and epsilon. The light chains are of 2 types: kappa and lambda. 1/4 of the amino terminus region of heavy chain is variable. The remaining 3/4 is constant. The amino acid sequence of variable regions of light chain and heavy chain is responsible for the specific binding of immunoglobulin with antigen. There are certain hypervariable regions within the variable region of light chain and heavy chain. The light chains have 3 hypervariable regions. Heavy chains have 4 hypervariable regions. The hypervariable region more specifically determines the antigen binding site. The composition, the composed of a single chain. The major immunoglobulin present in the highest among the plasma. They produce in response to the various infections and protects the body against infection. IgG can cross the placenta from the mother's blood to the fetus and provides immunity to the fetus. Triggers the foreign cell destruction mediated by complement system. IgA occurs as a single or double unit held together by J chain. They are produced by secretory cells of the respiratory tract, digestive tract, urinary tract, and is present in the mucus secretion of the cells. It prevents the entry of bacteria into the body through the cells. IgM, the largest immunoglobulin, composed of a 5Y shaped unit held together by J polypeptide chain. They cannot transverse blood vessels, hence it is restricted to bloodstream. It is the first antibody to be produced whenever a bacteria or viruses attacks the body. It is also produced in fetal stage itself. It is composed of single Y-shaped monomers, IgE molecules tightly bind with the mast cells, which releases histamine and causes allergy. It is produced by the plasma cells of respiratory rate. It increases the allergic diseases. Hyper gamma globin anemia increases serum gamma globulin level is called as hyper gamma globulinemia. Increased gamma globulin level is seen in following conditions: infection from the foreign bodies, chronic liver disease. This is a generalized increase in gamma globulin on ultrafluorescence. Gamma globulin gives a diffused but a deep stained band. High multiple myeloma, Waldenstrom, hyper gamma globulinemia increase in IgM only. Multiple myeloma, this is a malignant disease of plasma cells. It is in this case one type of plasma cells multiplies abnormally and produces to one type of immunoglobulin in excessive quantities, and other immunoglobulin or decrease. The ultrafluorescence of such a serum shows a thick, deeply stained protein band in the gamma globulin. This band is called as M band. This type of increase is known as a monoclonal gammopathy. Biochemical findings. The biochemical findings of multiple myeloma are increased total protein and decrease albumin, increase globulin and CA2 plus level in plasma. Some cases of multiple myeloma patients excrete light chains of immunoglobulin in urine, which are known as Ben-Jones protein. One type of immunoglobulin is increased in excess and other type is decreased. Hypogamma globulin, the plasma globulin level or decrease in the following condition: congenital defect in the synthesis of gamma globulin, leukemia, protein losing disease like a nephrotic syndrome. Estimation of IgG, IgA, IgM. The individual estimation of immunoglobulin is done by immunoassay methods such as using specific anti-serum radioimmunoassay method and immunoturbidometric method. Proteins in urine. Normal urine contains a very small amount of proteins. This is because a normal glomerulus does not allow high molecular weight proteins, including albumin, to filter. Only a low molecular weight proteins are allowed to pass through it. The proteins which are present in urine normally are alpha and beta globulin of low molecular weight. Protein in urine in disease conditions. Increased concentration of protein in urea, the norm is called proteinuria. The protein which are seen in urine may be albumin, hemoglobin, myoglobin, depending upon the state of disease. Albumin is the most common protein which is excreted in the urine under disease conditions. The pathology condition associated with albumin in urine or nephrotic syndrome and glomerulonephritis. In both cases, there is a damage to glomerular membrane causing protein to filtrate into the urine. Physiology condition where albumin is excreted or pregnancy or orthostatic proteinuria. Blood and HB. Excretion of blood in urine is known as hematuria. When renal tubule, bladder or glomeruli or severely damaged blood is passed into the urine. Presence of free hemoglobin without RBC is known as hemoglobinuria. Hemoglobin is mainly excreted in the urine when there is a severe intravascular hemolysis, mainly in mismatched blood transfusion or some infectious disease. Myoglobin is a protein formed in muscle cells. Its structure is similar to that of hemoglobin, which is present in muscle cells, and it is found in urine when there is a severe muscle cell breakdown. Quantitative test for urine protein heat and Benzoids protein are the light chain fragment of immunoglobulin which are excreted in the urine. In some cases of multiple myeloma, it was discovered in the urine by the characteristic behavior on heating. Benzoids protein precipitated between 40 degrees to 60 degrees centigrade. But as the temperature increases above 60 degrees centigrade, the protein dissolves. Again, on cooling, the protein gets precipitated. Protein in CSF. The cerebrospinal fluid is produced. CSF is produced in ventricles of the brain and passes on to the space around the spinal cord. The spinal cord ends near the first lumbar and fluid accumulates below. This is known as a lumbar fluid. This is a portion of CSF which is examined in routine cases. It is obtained by passing the needle between the L3 and L4 The total volume of CSF is 130 ml. Normal lumbar CSF has been followed characteristic. Protein concentration 15 to 45 milligram per deciliter. Glucose concentration is about 20 milligram less than protein plasma. Plasma glucose chloride is 120 to 130. Color reactions of a protein. The proteins are made up of amino acids joined by the peptide bond. So peptide bond and the different amino acids present in the proteins gives color product when they react with a variety of reagents. These tests are very important in the direction of amino acids in the biological field. Some of the tests are useful in the quantitative determination of protein and amino acids. Collectively, these tests are called as color reactions of protein. A few tests are given below. Bioroot test, inherent test, xanthoprotic acid, Melion test, aldehyde test, Sakaguchi's test, sulfur test, poly test, Mochi's test.